Lecture 1, Introduction to Computational Biology and Bioinformation. Welcome to this course on computation biology and bioinformation. This field merges biology, computer science, and mathematics to analyze, interpret, and visualize complex biological data. In today's era of genomics and big data, computational biology plays a central role in drug discovery, disease diagnosis, agriculture, and personalized medicine. In this module, we will study the molecular basis of life, DNA replication, transcription, and translation, genome organization and molecular biology techniques, DNA and protein databases, sequence retrieval and alignment, restriction maps, dot plots, gene prediction, phylogenetic trees and evolutionary analysis. Let's begin by revisiting the fundamentals of molecular biology. Lecture 2, DNA replication, transcription, and translation. 1. DNA replication. DNA replication is the biological process by which a cell copies its DNA before division. It is semi-conservative, meaning each new DNA molecule has one old strand and one new strand, initiated at specific regions called origins of replication. Helicase unwinds the DNA double helix. DNA polymerase synthesizes the new strand in the 5'3" direction. Primase lays down RNA primers. Ligase seals the gaps between Okazaki fragments on the lagging strand. 2. Transcription. Transcription is the process of creating an RNA copy of a gene. RNA polymerase binds to the promoter, synthesizes a strand of messenger RNA, mRNA. mRNA undergoes splicing, capping, and polyadenylation. 3. Translation. Translation converts mRNA into protein. Occurs in the ribosome. tRNA molecules bring amino acids. Ribosome reads mRNA codons and forms a polypeptide chain. Together, these processes are part of the central dogma of molecular biology, DNA-RNA protein. Lecture 3, Genome Organization and Molecular Biology Methods. 1. Genome organization. The genome is the complete set of DNA in an organism. It includes coding regions, genes, non-coding DNA, introns, regulatory sequences, repetitive DNA. In eukaryotes, DNA is wrapped around histone proteins forming chromatin. Genes are often organized into exons and introns. 2. Molecular biology methods. PCR, polymerase chain reaction amplifies DNA fragments. Gel electrophoresis, separates DNA based on size. DNA sequencing, e.g., Sanger, next-gen determines base sequence. Southern, northern, western blotting, detect DNA, RNA, and proteins respectively. CRISPR-Cas9, gene editing tool. These methods generate the raw data that computational tools analyze. Lecture 4, Introduction to DNA and Protein Databases. To store and access biological data, specialized databases have been created. 1. What are biological databases? Databases are repositories that store nucleotide, protein, expression, and structure data. 2. DNA databases. GenBank, NCBI Comprehensive Nucleotide Sequence Database. EMBLEBI, Europe offers sequence and functional data. DDBJ, Japan part of the INSDC collaboration. 3. Protein databases. UniProt KB, high-quality functional protein info. PIR, SwissProt, curated protein sequences. PDB, Protein Data Bank 3D protein structures. 4. File formats. FASTA, for sequence data. GenBank format, rich annotations. PDB format, 3D coordinate data. These databases support information retrieval, querying, and computational analysis. Lecture 5, Database Queries and Sequence Retrieval. To extract specific biological information, users submit queries to databases. 1. Search techniques. Keyword search, gene name, accession number. Sequence search, use tools like BLAST to find similar sequences. Field-specific search, e.g., organism, gene function, length. 2. Sequence retrieval. Faster format retrieval. Accession numbers used to track entries. Downloading sequences for further analysis. 3. Bioinformatics tools for querying. NCBI Entres, integrated search across multiple databases. EBI tools, sequence fetch, translation, alignment. Efficient data retrieval forms the basis for comparative and evolutionary studies. Lecture 6, Restriction maps and dot plots. 1. Restriction endonuclease mapping. This involves identifying the sites where restriction enzymes cut DNA. Enzymes recognize specific palindromic sequences. Maps help design cloning strategies. Tools, ENIPCutter, WebCutter, BioEdit. 2. Dot plots. Dot plots visually compare sequences to detect regions of similarity, repeats, and inversions. X-axis versus Y-axis of two sequences. Dots indicate matching segments. Tools, Dotter, Emboss dot matcher. These tools offer insight into genomic rearrangements, duplications, and conserved regions. Lecture 7, Sequence alignment, local, global, and multiple. Sequence alignment compares DNA or protein sequences to find regions of similarity. 1. Local alignment. Identifies similar regions within two sequences. Uses Smith-Waterman algorithm. Suitable for detecting motifs or conserved domains. 2. Global alignment. Aligns sequences from start to end. Uses Needleman-Wunsch algorithm. Ideal for comparing closely related sequences. 3. Multiple sequence alignment, MSA. Aligns more than two sequences. Reveals evolutionary relationships, conserved sites. Tools, Cluster Omega, Muscle, T-Coffee. MSA is essential for constructing phylogenetic trees and identifying functionally important residues. Lecture 8, Alignment scores and statistical significance. Alignment algorithms return scores that reflect the quality of the match. 1. Scoring systems. Match score, reward for identical bases. Mismatch penalty, subtracted for mismatches. Gap penalty, for insertions or deletions. 2. Statistical significance. Not all high scores are meaningful, random matches are possible. E-value, expect value probability that the match occurred by chance. Lower E-values indicate more significant matches. Tools like BLAST compute this automatically and provide graphical output. Lecture 9, Genetic distances and phylogenetic tree construction. Phylogenetics explores the evolutionary history of genes or species. 1. Genetic distance. Measures how different two sequences are. Calculated using alignment scores. Models, Jukes-Cantor, Kimura, Tomura NEI. 2. Distance-based phylogenies. UPGMA, unweighted pair group method assumes constant rate of evolution. Neighbor joining, NJ flexible and widely used. 3. Tree construction tools. Mega, PHYML, RXML, FastTree. Tree diagrams help visualize common ancestry and divergence times. Lecture 10, Character-based phylogenies and consensus sequences. 1. Character-based phylogenies. Use discrete characters, e.g., nucleotide positions, to build trees. Methods, maximum parsimony, maximum likelihood. 2. Consensus sequences. A consensus sequence represents the most common residues at each position in a multiple alignment. Helps define motifs. Used in primer design, functional prediction. Consensus sequences simplify complex data and highlight biologically relevant features. White Heavy Checkmark Module 1 Summary. In this module, we covered 1. The molecular processes of DNA replication, transcription, and translation. 2. Genome organization and key molecular biology methods. 3. The use and structure of DNA and protein databases. 4. Techniques for retrieving sequences and creating restriction maps. 5. Fundamentals of sequence alignment and assessing alignment quality. 6. Phylogenetic methods for understanding evolutionary relationships. This foundation prepares you for Module 2, where we'll focus on advanced topics such as microarray analysis, transcriptomics, functional genomics, proteomics, and database tools like BLAST, FASTA, and protein data banks.